Hi everyone, my name is Paul Riggs, and today I'll be talking about my assigned paper, Global Diversity and Biogeography of Bacterial Communities in Wastewater Treatment Plants. This study, conducted by Wu and their colleagues from the Global Water Microbiome Consortium, examines what kinds of microbes live in wastewater treatment plants around the world, how those microbial communities differ and resemble one another, and how they contribute to overall plant function. Wastewater treatment plants are one of the most critical engineered ecosystems on Earth. Yet, until recently, we didn't really understand the microbes that make them work on a global scale. What makes this paper especially important is that it's the first worldwide survey of these microbial communities, and it revealed the bacterial diversity inside wastewater plants is nearly on the same order of magnitude as the entire ocean's microbiome. That discovery completely changed how we think about these systems. They're not just industrial processes, but are incredibly diverse and dynamic ecosystems. To understand the microbial makeup of wastewater treatment plants on a global scale, the researchers collected from 269 wastewater treatment plants across 23 countries and 6 continents, adding up to about 1,200 activated sludge samples. The map here really highlights how extensive their sampling was, covering nearly every major climate zone and giving us the first truly global picture of wastewater microbiome. With the samples, the researchers aim to 1. Measure the global bacterial diversity in wastewater plants, 2. Identify if a shared core microbiome exists worldwide, 3. Test if diversity changes with latitude, 4. Link diversity to treatment performance, and finally 5. Determine if communities are shaped by environment or chance. When the researchers analyzed all the samples they gathered, they found huge diversity, about 1 billion unique bacterial types, but comprised only of a small global core. Only 28 core operational taxonomic units were found across every plant. That's just 0.05% of all detected types, yet together they make up about 12% of all the total abundance. So while each plant has its own local makeup, these few species dominated everywhere. Most of them belong to proteobacteria, especially beta proteobacteria as shown in the purple bar to the right, and also include some familiar and functionally important groups such as Nitrospira, which oxidize nitrate during nitrification, Zooglobacter, which help flock particles stick together, and Accumulobacter, which store phosphorus in EBPR systems. These findings shown in figures 2a and 2b illustrate that even though the core is tiny, it plays a major role in treatment performance. So even though the same core microbes show up everywhere, the overall diversity still varies from place to place. Interestingly, the study found no clear north-south or latitudinal pattern in the microbial diversity, which was unexpected because in most ecosystems, like forests or oceans, diversity usually increases towards the equator. Instead, here, richness actually peaked at mid-latitudes. If you look at figure 4.1d, you can see that the highest species richness occurs in those mid-latitude regions, with diversity dropping slightly towards the poles and the equator. The same figure also shows how temperature tracks closely with changes in diversity. Among all the deterministic or environmental factors, temperature had the strongest influence on which microbes are present, even more important than latitude, continent, or plant design. So warmer or colder operating conditions shift the community structure more than simple geography does. But it's important to note that this only explains part of the picture. While temperature is the dominant environmental driver, the researchers also found that random or stochastic processes, things like dispersal, drift, and chance colonization, still account for the majority share of the variation. That idea of randomness shaping diversity is what we'll look at next. So after identifying temperature as the strongest environmental factor, the researchers looked deeper into what actually drives these global patterns in microbial diversity. In fact, they found that both deterministic and stochastic processes are at work. Deterministic factors include things like temperature, in-fluid composition, solid retention time, dissolved oxygen, and hydraulic retention time. These are predictable and measurable variables that clearly shape which microbes live in a plant. But randomness still plays a huge role. Microbes move around, they disperse, they drift, and sometimes they just get lucky when they colonize a new system. If you look at the figures on the left, you can see how environmental factors explain a portion of these variables, but over half remain unexplained. That tells us that chance and dispersal still have a major influence on how these microbial communities are assembled, even in highly engineered systems like wastewater treatment plants. Wu and their team also discuss a phenomenon called distance decay, where the farther apart two plants are, the more their microbial communities differ. However, they determined that this phenomenon is weaker than what we would see in natural ecosystems, probably because wastewater plants around the world share such similar operating conditions. To wrap everything up, here are the main takeaways from this study and what they mean moving forward. First, there's huge global diversity in wastewater treatment plants, roughly 1 billion unique bacterial types, yet all of it is anchored just by 28 core bacteria found worldwide. Second, those core microbes are closely linked to nutrient removal and overall treatment performance. They are the reliable workhorses that keep these systems running efficiently and effectively everywhere. Third, microbial communities are shaped by both environmental factors and chance. Temperature and operating conditions play major roles, but random dispersal and ecological drift also influence who thrives. Fourth, there is no clear latitudinal pattern diversity. Richness peaks at mid-latitudes, likely due to temperature and plant operation rather than geography. And finally, there is strong functional redundancy. Different species often perform the same roles, keeping treatment plants stable and efficient despite community differences. And moving forward, this study shows how global ecology can guide better engineering. Tracking core microbiomes can help monitor and improve performance. Applying ecological principles can make systems more resilient and adaptable. Global data can guide local optimization by showing what works better under similar conditions. And managing key factors like temperature, solid retention time, and oxygen can favor the microbes that drive nutrient removal and stability. Altogether, this study reframes wastewater treatment as global microbial ecosystems that follow the same ecological rules as nature, reminding us that understanding biology is key to building efficient and suitable systems. Thanks for listening.